Lipids. What are the two major roles of lipids in your body? Long-term energy storage and yeah, what I was thinking about those insulation, keep you warm, help you maintain your body temperature, but also things like steroids, cell membranes are made out of lipids. There's lots of functions of lipids. This lipid right here that we're looking at is a triglyceride, the most basic kind of lipid. It's called a triglyceride because it's built around this thing, which is glycerol, and attached to it are three, hence the name tri, fatty acids. It's really good at storing energy because it has a lot of what? Has a lot of bonds, and bonds are the places where energy gets stored in chemicals. It's a triglyceride. Now, if we wanted to make this a diagram of a phospholipid, what's the only thing we would need to change? Get rid of one of these, and instead attach what? A phosphate group. And that phosphate group will make this side be what? Polar. These sides are basically just carbons and hydrogen, so they're going to be non-polar. And therefore we refer to this kind of molecule as amphipathic. If we put those things in water, those phospholipids, we know they're going to form a what? A bilayer. And remember, this diagram that we see of phospholipid bilayers looks linear. That's not a straight line. It's really going to be what? A sphere. Okay, not just a circle, a three-dimensional sphere where the heads are always pointed toward the water and the tails are always pointed toward each other, away from water, right? These are inside. These are outside and the very inside, pointing toward the water inside the cell. But backtracking just a second on the lipids. You also need to know the difference between a saturated and an unsaturated fat. Chemically, basically the only difference is unsaturated fats have what that saturated ones don't. Some double bonds between carbons. And wherever there's a double bond, what does that cause to happen to that chain? It kinks, it bends, which makes these things a lot more likely to be what state of matter? Liquids and these solids, because they stack up really tightly. And they can solidify where? In your coronary arteries and lead to atherosclerosis, heart disease, clogging of the arteries. These don't do that very often. Now, I'm not saying you should eat a whole lot of this either, right? Because it's really high in like calories and it's going to make you get fat. But these are worse for your heart than these are. What now?
I'd rather have fat. I'd rather get fat.
Oh yeah. Well, these are going to make you fat and die. These are just going to make you fat, right? They both have a lot of calories. Remember, if you see 4 carbon rings fused together, that's going to be steroid. 3 steroids you ought to know the name of. Estrogen, testosterone, and cholesterol. If you see this name right here, that is another name for estrogen, estradiol. So don't let that confuse you, that's just estrogen. Anytime you see that estra, that's going to have something to do with the female menstrual cycle. But these are steroids. Cholesterol is found where in your body? It's in the cell membrane. It helps to make it a little bit thicker, a little bit less liquidy. And it's also a precursor. It's a building block for the other steroids. To make testosterone, guys need some cholesterol.
Is cholesterol what we were talking about, the rebar?
No, that's the cytoskeleton stuff. This is the protein.
Now what's that called?
Cytoskeleton. Yeah.
So guys with higher testosterone could potentially have higher cholesterol?
I don't think there's a one-to-one connection there. I don't think so. Because it turns out if you don't eat cholesterol, your body will make it. Your body can generate it. There are some people with a genetic disease called hypercholesterolemia. That's pretty descriptive of what it is. It just means what? Yeah, really high cholesterol. They might not eat, anything bad. They've got high cholesterol because why? They're making it. And they end up, the people with that disorder end up having heart attacks sometimes in their 20s because they're making all this cholesterol that's clogging their arteries. Well, that's, what do they treat high cholesterol with, guys? No, you guys should know this because I bet some of your parents are on it.
Like Bayer, aspirin.
How do they treat high cholesterol? You guys should know this. They commercial it on TV all the time. Lipitor, right? You all seen those commercials for Lipitor? That's what Lipitor is medicines are. They're called statins.
Statins was one of them male things.
No. Lipitor is not like Viagra. No. Lipitor is about lowering cholesterol. Viagra is about other things. We'll just leave it at that. No, not going into that.
There's another one.
I'll say Google it if you want to know. Google it.
There's
Yes.
another one I'm confused with Lipitor. Or one of them antidepressants or something like that.
Lunesta is like an antidepressant, sleep medicine kind of thing.
Yeah.
There's a ton of them. But anyway, today we want to focus on proteins. Most important chemicals in your body. You guys should already know they're made of these four things. Always. We know that from that diagram over there that you all drew on your quiz and you all got right? So you know those things are in there. Sometimes there's some other elements. Particularly, what other element's pretty common in proteins? Sulfur. We'll talk about sulfur in a minute. It's not in everyone, but it's in most proteins. Sulfur is. If I ask you what are the uses of proteins in the body, in life, what would be a good answer? Everything. Proteins make up the structure of everything, and they make up your enzymes, so they also control what? Your chemical reactions, your functions. So pretty much everything. When you hear protein, though, the first thing I want you to think about is enzymes. Enzymes are your body's most important proteins. They make you work. You all know, hopefully, that amino acids are the monomers that make up proteins. They're the pieces, building blocks. And in nature, there's only 20 amino acids found in living things. Those 20 pieces make up every living thing, from bacteria, to protists, to fungus, to plants, to animals. 20 different building blocks for life. You've got to be able to draw an amino acid. Remembering that all of that is exactly the same in every amino acid. The part that's different is this part down at the bottom. So let's look at a couple of pictures.
So, amino acids found in living things, does that mean like 20 different ones?
20 different kinds of amino acids. Remember, you might see amino acids in their non-ionized form, which is what we drew. It's just as right to draw it this way, because what do we know about that hydrogen? It can break off, become acidic, and when you lose an H+, whatever's left has to become negative, right? This amino group is also what kind of chemical? It's a base, which means it takes in hydrogens. So sometimes you'll see it drawn that way. If you gain an H+, you become plus. But realize these are the same thing. This just lost an H, that gained an H. Same thing. Be able to draw an amino acid. Now here's some diagrams of some different amino acids. First thing you ought to notice is pretty much all their names end in INE. In this class, if you see a chemical whose name ends in INE, it's probably an amino acid. There's some exceptions like glutamate, but most of them end in INE. Notice that the parts that I'm circling up here are exactly the same in every one of them. But also notice the categories that they're divided into. These top ones are called nonpolar. Because if you look at their R groups, what's not in those R groups? Not in oxygen or nitrogen. Which in biology, those are the two elements you need to worry about being what? Well, they're electronegative, which causes things to be polar, right? Do we really need to worry too much about fluorine and chlorine in biology? No, not going to find those in the body much, right? Chlorine ions you'll find. But in terms of proteins, the two things that might make them be polar, nitrogen and oxygen, these don't have them. But notice if you drop down to this next category, they do. What's that functional group right there called? Hydroxyl group. If it's attached to something, that something that it's attached to is more than likely going to be a polar kind of thing. Let's talk about why that's important. Let's say that this amino acid right here, this black one, is one of these hydrophobic amino acids. That was the first one in the chain. Okay? I know. Let's say all these other ones are hydrophilic. One of these. So, hydrophobic, hydrophilic. This thing can't avoid being around water, right? If it's in a living thing, it's got to be around water. By the way, what would the name for this thing right now be? This is supposed to be a chain of amino acids. What's the name for that? A polypeptide. It's a polypeptide. It's not a protein yet because it hasn't what? It hasn't taken on its shape. It hasn't combined with other polypeptides. Right now it's just a polypeptide. When this thing goes into water, because this one's nonpolar, hydrophobic, and all the rest are polar, hydrophilic, what's going to happen when it gets into water? It's going to bend, twist, so that this black one, this hydrophobic one, is where? In the middle, as far away from what as possible. As far away from water as possible. It's hiding in there, right? That thing that I just described is one of the main reasons, one of the main determinants of what? The shape of a protein. Hydrophobic and hydrophilic R groups cause these amino acids to bend and twist into a certain pattern. And that helps to determine the shape of the protein. Hydrophobic ones are always going to bend toward the middle of the protein, away from water. Hydrophilic ones are going to bend toward the outside, near the water. That makes sense? And again, this is called a polypeptide, a chain of amino acids. What holds this amino acid to the one beside it, one beside it, one beside it, and so on. It's called a peptide bond. That bond, that peptide bond between amino acids, is what kind of bond? You learned about three main kinds of bonds in Chemistry 1. Metallic, covalent, and ionic. That's what? It's covalent. Because amino acids are made out of what? All what? What all those things have in common in that amino acid? They're nonmetals. Nonmetals form covalent bonds. So it's covalent. Peptide bonds are covalent. You need to understand how we get a covalent bond. So let me get an eraser here, a marker. Let's take this amino acid right here and hook it up to another amino acid and figure out how that works. So here's amino acid number 2. What do we call reactions in biology that hook together monomers to make polymers? Condensation or synthesis. And so the peptide bond is always going to form between what two parts? What two functional groups? Carboxyl to amino. And remember, it's a condensation reaction. Here's how that works. This OH right here is removed, freeing up what? Taking off that OH, freed up what? A bonding site, right? There's an open spot there. What has to be removed over here? One of those hydrogens are removed to open up the bonding site over there. Well, that H and that OH, they get together to make water. That's what's called what? Condensation. And now that bond forms between carboxyl on one amino acid and amino on the other. That is what we call a peptide bond. Make sense? All right, so what we're going to look at now is it's the last thing we'll get to today probably, and that's the structure of a protein. Structure, we know, determines function. The structure of a protein is organized on 4 levels. You're going to need to be able to explain each of those 4 levels. And it's primary, secondary, tertiary, quaternary. That's just a fancy way to say 1, 2, 3, 4, right? First level, second level, third level, fourth level. Let's start with level number 1, primary structure. The primary structure is very simple. It's what amino acids are in that protein and what order are those amino acids in. Which amino acids are in here? Out of the 20, which one goes first? Which one goes second? Which one goes third? Which one goes fourth? So it's which amino acids are in the protein, what order are those amino acids in. That's the primary structure. That's the only level of structure that what codes for. That's what your DNA codes for. That's all your DNA codes for. Primary structure of a protein. Put this amino acid number 1. Put this one number 2. Put this one number 3. Put this one number 4. That's what your DNA tells the ribosomes. This one goes first.
So does all DNA start out the same?
Does all, I don't get what you're saying.
Not DNA, it's all protein. Do all proteins start out the same until they change shape?
No, because they have different amino acids in a different order. Like one gene may say put methionine first and glutamate second and serine third. And that'll make a different protein than another gene that puts other amino acids in different order. Primary structure, what amino acid goes first? What amino acid goes second? What amino acid goes third? What amino acid goes fourth? It's the primary structure. Now again, what do we call a chain like this? Polypeptide. The second level of structure, secondary structure, let me just define it for you. It's the way certain segments of a single polypeptide are shaped. The way certain segments of a single polypeptide chain are shaped. Let me give you an idea of how this works. Let's say we put this thing in water and it all bent and twisted. Let's say this orange amino acid right here and this green one both have polar R groups. So when they bend and twist, they get close together. What happens when 2 polar things get close together? They're going to make what kind of bond? Hydrogen bond. They're going to stick. And let's say the same thing's going on over here. This black one and this orange one are both polar. They get together. They make a hydrogen bond. What does that mean about the shape once it's formed and those bonds are made? It's stuck that way, right? Those hydrogen bonds, their function is to hold the shape in place. When you hear secondary structure, I want you to think about hydrogen bonds. Hydrogen bonds, bonds between polar amino acids, help to hold the secondary structure in place. Hydrogen bonds hold secondary structure in place. Yes?
What did you say secondary was in?
It's the shape of individual segments of a single polypeptide. So this side over here may be shaped one way, and this side over here may be shaped a different way. This is one chain. The way different parts of a single polypeptide chain are shaped. A lot of times, proteins get shaped into a helix, kind of like DNA, spiral staircase. Sometimes they'll get like pleats on pants, so pleated sheets. They'll go up and down, up and down, up and down. But regardless, whichever shape it bends into, that shape is held in place by what? Hydrogen bonds between polar amino acids. Tertiary structure. Tertiary is third level. Tertiary is what's the shape of one entire polypeptide chain? What's the overall shape of an entire polypeptide chain? Maybe like that. What's the shape of the whole chain? What's the shape of an entire polypeptide chain? Now I want to backtrack for just a second. I want to point out to you a specific amino acid. This one turns out to be an important one. What's different about that amino acid, cysteine, and the others? It's got sulfur in it. What do we call that functional group right there? Sulfhydryl. Turns out that cysteines are really important for this reason. Let's say this thing bent and twisted into a certain shape. And what causes it to bend and twist that way? Some amino acids are hydrophilic and want to be where? Near water. Some are hydrophobic and want to be away from water. So it bends and twists mainly because of that. But let's say these two yellow ones, and why am I choosing yellow here? Why would yellow be an appropriate color to choose here? Sulfur's like bright yellow. You guys seen pure sulfur before? Bright yellow? No?
Pictures.
Alright, so in the back. I'll have to pull that. Anyway, sulfur's yellow. Those two cysteines get near each other, and what do? Not hydrogen bonds. The two sulfurs on each cysteine actually form this bridge called a disulfide bridge, or disulfide bond. It's stronger than what? Stronger than a hydrogen bond. When you hear tertiary structure, I want you to know it's the shape of the whole polypeptide, but I also want you to think about these disulfide bonds. They help to hold, once the tertiary structure forms, they help to what? Keep it there, hold it there. Hydrogen bonds also help to hold it there. So when you hear tertiary structure, I want you to think about disulfide bonds and hydrogen bonds holding it in place, keeping it that way, keeping it from what? Keeping it from changing, from denaturing. Because if it changes, it's not going to work anymore, right? Structure determines function. So disulfide bonds and hydrogen bonds hold the shape of the protein in place. And that leads us to our last level, which is called the quaternary structure. Key thing, quaternary structure is the overall shape of a protein. The overall shape of the entire protein. But here's what you got to understand to get that. A protein is usually not made out of just one of these polypeptides, right? It's made out of several polypeptides all tied or wrapped together. So polypeptide 1, polypeptide 2. The quaternary structure is how do these different polypeptide chains wrap around each other? How do multiple polypeptide chains wrap together to form what? The overall shape of the protein. Let me say that one more time, because that's a good definition of quaternary structure. How do multiple polypeptide chains wrap together to form the overall shape of a protein? Quaternary structure. How do multiple polypeptide chains wrap together to form the overall structure of a protein? 2 or 3 other quick points. We keep saying structure determines function. We know a change in the shape of a protein is what? Denaturation. It's an important word to know. And what 3 factors have we talked about that cause denaturation to happen?
pH, heat, and
What? Yes. pH, heat, and salinity. pH either way, right? More acidic, more basic, either one can cause it to happen. Temperature, we got to go which way to denature something. Got to hot. Got to be hot, because to denature we need to break bonds. And then salinity can go either way, more salty, less salty. What kind of bonds are we breaking when we change the shape of this protein? The hydrogen bonds and or the disulfide bonds. We're not breaking.
those are the only 2 bonds.
We're not breaking those guys, right? What are those guys called? We're not breaking the peptide bonds. When you denature a protein, you're breaking the hydrogen bonds and the disulfide bonds.
Those are the only 2 bonds we need to worry about.
Those, in terms of denaturation, yeah. And what? Hydrogen and disulfide. When you talk about denaturation, the protein is losing its tertiary structure and its secondary structure. That's really what we're changing, because that's where what's important. Those kinds of bonds, hydrogen and disulfide. So, to my circle, when you get denaturation, we're talking about a change in the secondary or tertiary structures of the protein. All right, I want to do two more things, and then I need to talk about your homework assignment. Here's what we just drew on the board over there. We've got two amino acids going through a condensation or synthesis reaction, remembering that the OH from the carboxyl group is removed, and H from the amino group is removed, and that forms a peptide bond right in here between carboxyl and amino. Sometimes this end of the protein right here is called the N-terminus because it's got an amino group on one end.
You need to know that.
Yep. The other end is called, guess what? The C-terminus. Because it has what? Carboxyl. The end back. Protein's usually going to start, always going to start with an N-terminus and end with a C-terminus. This picture just kind of reminded us of these structures. Remember, primary structure. What amino acids are in the protein and what order are they in? That's what's coded for by DNA, primary structure. Secondary structure, how are certain segments of each polypeptide chain shaped? A lot of times they'll be in a helix. Sometimes they'll be in pleated sheet. Regardless, what do these colon-like dots up here represent all over the place? Well, you can all read hydrogen bonds. When you think secondary structure, you should think hydrogen bonds because they hold it in place. They hold the secondary structure in place. Tertiary structure is the shape of each what? Each polypeptide chain, and that's held in place by what? Two forces. Hydrogen bonds and disulfide bonds, which we see up here looking like a streak of lightning or something.
Is it ever both, or is it and or?
It's both. Disulfide and hydrogen. I don't know of any exceptions. And then here's quaternary structure. Remember, the quaternary structure is the overall shape of a protein determined by how what happens? How multiple polypeptide chains wrap together. That determines the overall shape. All right, the last thing that I will mention today, and tomorrow, we got two things we need to finish before the test. We need to talk a little bit about nucleic acids, which is our fourth macromolecule. And then most of tomorrow is going to be about water, which is what your Moodle assignment you're about to turn in is about, right? Water, water, water. But nucleic acids, their functions are pretty diverse in the body. But the two that we think of first usually, DNA and RNA, are about storing genetic information, obviously. The other one you should think about next, ATP, is about storing energy. There are others. When we talk about cell signaling, cell communication in a few weeks, we're going to talk about this chemical called cyclical AMP, CAMP. That's also a nucleic acid. And there's others. But for right now, just worry about these three. Realize that, like pretty much all biological chemicals, they're big molecules, right? They're macromolecules. They're polymers. The monomers that make up nucleic acids are called nucleotides. In next class, we'll talk about what a nucleotide looks like and how it works. Let me stop the computer there.